Hello, I am Sam Smith, and this is Sam's Mcat Basics. This podcast covers the most important topics put out on the AAMC Mcat each year, and I determined this list by going through all the official practice materials that the AAMC puts out, and also some third-party practice material, and just put together this big list of topics that consistently kept on showing up, and ranked those topics and made podcasts out of it. This podcast covers topics within metabolism. It's geared towards reviewing the three most important metabolic processes, which are glycolysis, the Krebs cycle, and the electron transport chain. And I'm going to cover these three processes in detail, and then I'm going to go into kind of my philosophy on how I memorize the structures and the names within each of these processes, and kind of some of the stuff you need to memorize I'll go over. And it's interesting for me to see that a lot of the study materials I went through, specifically the Kaplan books, really didn't put much emphasis on this material, but then it kept showing up on a lot of the practice tests I was taking, the official practice tests I was taking, and the question packs and stuff. And so for me, I think it was a pretty important topic, and this definitely did come up quite a bit. So you'll see this kind of material on one out of the four MCAT sections, and that is the biological and biochemical foundations of living systems. Enjoy. Hope it helps. So, I just want to start by saying that this subject material is very dense. You might have to listen to this a few times, let it soak in, think about it, pause it, rewind it, whatever you need to do. Just know that it's important. You're going to come across a lot of enzyme names, intermediate names, products, reactant names, stuff like that. It's just more memorization than it is conceptual. So, yeah, just let this stuff sink in. Make sure you know it. All right, so I'm going to start this lecture on metabolism with the definition of metabolism. So metabolism is defined as the set of life-sustaining chemical reactions that occur within an organism. So that's a pretty broad definition. However, I'm going to focus on glycolysis, Krebs cycle, electron transport chain, and anaerobic respiration, as well as talk a little bit about where each one takes place. So to start, let's get into glycolysis. Glycolysis is the first step in the breakdown of glucose, and it occurs in the cytosol. And essentially what's going on is, in the overall reaction, you are splitting glucose into two 3-carbon molecules called pyruvate. And it's a pretty old metabolic pathway, and I just mean that it evolved a long time ago. And we know this because it is found in a great majority of organisms that are alive today. And overall, the reaction has 10 steps. And if you really want to, kill the MCAT, know all of them, know everything about each one. But what I'm going to do is I'm going to go over how I kind of memorized it and how I thought about it. That really helped me memorize each component. You'll see what I'm saying when I go over this. So what I'm going to do first is go through all the intermediates as well as the enzymes, and then once I go through those, I'll explain how I memorize them. So in step number 1 in glycolysis, glucose is converted to glucose 6-phosphate by hexokinase. In step number 2, glucose 6-phosphate is then converted to fructose 6-phosphate by phosphoglucose isomerase. In step 3, you have fructose 6-phosphate, which is converted into fructose 1,6-bisphosphate by phosphofructokinase. And in step 4, fructose 1,6-bisphosphate is broken down into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate by aldolase. And then once you have that dihydroxyacetone phosphate, which is one of the molecules that fructose 1,6-bisphosphate was broken down into, you now must convert that to glyceraldehyde 3-phosphate. So in step 5, dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate by triose phosphate isomerase. And then moving on, step 6, glyceraldehyde 3-phosphate, which now you have 2 of, are converted to 1,3-bisphosphoglycerate by glyceraldehyde phosphate dehydrogenase. And then in step 7, the 1,3-bisphosphoglycerate, which again you have 2 of them, is converted to 3-phosphoglycerate by phosphoglycerate kinase. And then those two 3-phosphoglycerates are converted to 2-phosphoglycerates. And that is done by phosphoglycerate mutase. And then those two 2-phosphoglycerates are converted to 2, again, phosphoenol pyruvates by enolase. And then in the last step, your 2 phosphoenol pyruvates are converted to 2 pyruvates by pyruvate kinase. And again, that is a bunch of shit to memorize. So what I did is I broke it up into 3 different areas. First, I memorized a simplified version of this process. Then second, I memorized the rate-limiting enzymes. And lastly, I memorized the energy-using and energy-producing steps. And I'm bringing these up because I think these are the 3 most important parts of glycolysis for you to know for the MCAT. All right, so the first thing that is important to remember is the simplified process. So what's really going on to this glucose? Well, step number 1, you add a phosphate to glucose. And what does that do? That activates it. So step number 1, add phosphate to glucose. Step number 2, convert to fructose. And then step number 3, add another phosphate on. Step number 4, break that apart into 2 different molecules. Step number 5, add a phosphate to each. Step number 6, remove that phosphate. Step number 7, isomerize. Step number 8, convert to a pyruvate precursor. Step number 9, convert to pyruvate. So I basically memorized that kind of in my head. I think it's an easier picture to get than saying, okay, glucose was converted to glucose 6-phosphate by hexokinase. I mean, I guess that's kind of a shitty example because that is easy to remember. But what about, so like, to me that's easier than saying fructose 1,6-bisphosphate is converted to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. If I can just remember that, okay, well, what happens again? Oh yeah, so the glucose that has just been activated by that phosphate group was converted to a fructose, and then another phosphate group is added on it, and then that was broken up into 2 different molecules. Okay, well, in that way, I can essentially remember the steps of glycolysis and then reference each of those steps. So this kind of helped me keep everything straight in my mind. So I'm going to give you an example of kind of a way that I use this. So on the MCAT, they might give you a drawing of one molecule and, an arrow and then another molecule and say, in this glycolysis reaction, what enzyme catalyzes this reaction, and then you're going to just have the structures. So what I was able to do then is was able to go through that simplified process and just say, okay, this converts there, okay, that structure must be there, this structure must be there, and then from there I was able to say, kind of pick out an enzyme. And so to be able to pick out an enzyme, it helps to understand what enzymes do, and so that's going to be another podcast. But for example, an isomerase converts between isomers of different molecules, and so you can remember that. A kinase has a specific function, a phosphatase has a specific function, a dehydrogenase has a specific function, and so you're going to have to know that anyways. And so I would refer to the enzymes podcast, or the podcast that talks a little bit more about enzymes for this. But again, having that simplified process memorized really helped me. It's also important to note that you just have to be able to recognize these names. So the Mcat, if you don't know, is a fully multiple choice test. So it's a lot easier to obviously to recognize a name than to have to just write that name down word for word, letter for letter. So keep that in mind. All right, so the second most important thing to have memorized for glycolysis on the Mcat are the rate-limiting enzymes. So rate-limiting enzymes are unidirectional. That means that they catalyze the forward reaction, but they do not catalyze the reverse reaction in any appreciable amount. So remember that these are non-reversible reactions, and they can only go in one direction. So in glycolysis, these are hexokinase and glucokinase, phosphofructokinase, and pyruvate kinase. So again, hexokinase and glucokinase function to add a phosphate group to glucose. Phosphofructokinase converts fructose 6-phosphate to fructose 1,6-bisphosphate by adding a phosphate group. And lastly, pyruvate kinase essentially removes a phosphate group from phosphoenolpyruvate to make pyruvate. And so there is a little bit of a cheesy mnemonic that goes with the irreversible enzymes of glycolysis, and that is the following: how glycolysis pushes forward the process, kinases. So you have HGPFPK. So the H goes with hexokinase, the G with glucokinase, the P and the F and pushes forward goes with phosphofructokinase 1, also known as PFK, and the P and the K go with pyruvate kinase. I think it's kind of a stupid mnemonic. I would just straight up memorize them. You don't really need a mnemonic for those. There's only 4 of them. And number 3, most important thing to know, is the steps in which energy is produced. And so this is either produced in the form of ATP or NADH. So again, this goes back to knowing your enzymes. Everything that ends in kinase creates ATP or uses ATP, and the things that end in dehydrogenase create NADH. So overall, the glycolysis reaction is that glucose is converted to 2-pyruvate, 2-ATP, and 2-NADH. All right, now that I did a brief overview of glycolysis, I want to go into the regulation of glycolysis. What are the things that turn up glycolysis? What are the things that turn down glycolysis? How does that all work? So the first regulation I want to talk about is allosteric regulation of glycolysis. And allosteric regulation refers to regulation via a effector molecule binding at a site other than the active site on an enzyme. And in glycolysis, allosteric regulation affects the irreversible enzymes that I just talked about. So hexokinase is inhibited by glucose 6-phosphate, which is its product, and that essentially comes back and binds to the allosteric site and inhibits hexokinase. And then phosphofructokinase, on the other hand, is inhibited by ATP, which is the high-energy molecule that the body uses for energy, and also inhibited by citrate. And that feeds back from the Krebs cycle, and we'll get into that a little bit more. And then phosphofructokinase is activated by AMP, which is basically the low-energy form of ATP. All the energy in ATP has been used up. It's AMP, adenosine monophosphate. And then phosphofructokinase is also activated by fructose 2,6-bisphosphate. Now, you might be thinking to yourself, all right, what is fructose 2,6-bisphosphate? I haven't heard of that. Well, fructose 2,6-bisphosphate is a very potent allosteric activator of the phosphofructokinase enzyme. So other than that, don't really think about it too much. Just know that it really likes to get in there inside the enzyme, bind to the allosteric site, and then increase its activity. And lastly, the enzyme pyruvate kinase is inhibited by ATP and is activated by fructose 1,6-bisphosphate, which is part of the glycolysis. It's an intermediate in glycolysis. And don't mix up fructose 2,6-bisphosphate and fructose 1,6. Fructose 2,6 again activates phosphofructokinase. So now that I've covered glycolysis, I'm going to go into what happens next. So if oxygen is unavailable, i.e. you are under anaerobic conditions, glycolysis becomes the main form or the main way to create ATP. So your body keeps trying to use glycolysis to produce ATP. Well, what it has to do then is it has to regenerate NAD+. In one of the steps in glycolysis, the step in which you convert glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, you create an NADH. And so that NADH is only useful for energy if you have oxygen later. So what your body then needs to do is create more NAD+ so that you can convert that glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. Without that NAD+, this reaction will not go forward, and then you're missing out on some of the ATP that you could create. So, how does your body then regenerate that NAD+? So what your body does is it actually converts pyruvate into lactate. And then what happens with lactate is it essentially just drips straight out of your nipples, and you expel all that NAD+. No, just kidding. That's not what happens. What actually happens is your body takes pyruvate and converts the pyruvate to lactate, and in this process oxidizes NADH back to NAD+. And then you can use that NAD+ to go through glycolysis again, create more ATP, and so forth. And so, as you can imagine, that lactate builds up, and this can cause acidosis, or essentially where your blood pH gets too low. And you may have heard that the buildup of lactic acid is what makes you sore, but that's really just bro science. What is actually happening is you are getting micro tears or microscopic damage in your muscle fibers after you lift or work out, etc. And that's actually what causes soreness.
To reiterate my point, if oxygen is available, the body must regenerate NAD+ to keep glycolysis moving. So to do that, the body converts pyruvate, which is the end product of glycolysis, to lactate, and in the process regenerates NAD+ from NADH.
Now let's say oxygen is available. Well, then you can further oxidize the pyruvate that you created in glycolysis and strip even more energy, create even more ATP off that. So the next step in this process is called the Krebs cycle. So the overall goal of the Krebs cycle is to convert acetyl-CoA to oxaloacetate. And in this process, it gives off lots of high energy molecules that can later be converted to ATP.
Or are GTP themselves, which is like ATP, high energy molecule.
And this occurs in the mitochondrial matrix, which is the insidemost part of the mitochondria. So you might be saying to yourself, okay, didn't we just end glycolysis with the pyruvate? How did we get acetyl-CoA? Well, the pyruvate dehydrogenase complex is a complex of 3 enzymes, and it converts pyruvate to acetyl-CoA. And so it's got 3 distinct enzyme portions to it, which are E1, E2, and E3. And you should know the prosthetic groups that go along with each of these enzyme substructures. So I'm going to go through those. So for E1, the prosthetic group is thiamine pyrophosphate. For E2, the prosthetic group is lipoamide. And for E3, you have a few cofactors, and they are FAD and NAD+. All right, so again, I'm going to go over the process, or like I did for glycolysis, I'm going to go over the whole process. I'm going to tell you all the enzymes and all the intermediates. Then I'm going to go over a better way to memorize them, or more efficient at least. Maybe not necessarily better, but how I did it and how I feel is the most efficient way. All right, so starting with acetyl-CoA. So acetyl-CoA is converted to citrate by citrate synthase, which removes the CoA group from the acetyl group. And then citrate is further processed to cis-aconitate by aconitase. And then cis-aconitate is converted to deisocitrate, again by aconitase. And then isocitrate is converted to alpha-ketoglutarate by isocitrate dehydrogenase. Alpha-ketoglutarate is then converted to succinyl-CoA by alpha-ketoglutarate dehydrogenase. And then succinyl-CoA is converted to succinate by succinyl-CoA synthetase. And then succinate is converted to fumarate by succinate dehydrogenase. And then fumarate is converted to malate by fumarase. And lastly, malate is converted to oxaloacetate by malate dehydrogenase. So there is a very nice mnemonic in which you can memorize these intermediates. And before I give you that, let me note that this mnemonic does not take into account cis-aconitate, which I'm assuming is not that important. I would just not really worry too much about that. So the intermediates, you can remember the intermediates by remembering this phrase: please, can I keep selling seashells for money, officer? Again, it's quite the tongue twister, but it is, please, can I keep selling seashells for money, officer? And so the P, so it is PCIKSSFMO. And so that's pyruvate, citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. And so once you memorize these intermediates, it's pretty easy to remember, or at least to recognize the enzyme names, because these enzymes always refer to the name of the reactant that they're converting. So for example, succinyl-CoA synthetase converts succinyl-CoA to succinate. So, just remember that the name of the enzyme corresponds to the reactant in the citric acid cycle, also called the Krebs cycle. And of course, as always, it's important to know what these enzymes do. Do they produce energy? Do they not produce energy? What exactly are they doing? So, and this again goes back to just knowing what enzymes do in general, which is in another podcast, but I'm going to go over it slightly here right now. So dehydrogenases transfer a hydride group to an electron acceptor, and these typically reduce NAD plus or FADH. And synthases do not require energy to form bonds, so these form covalent bonds but don't use up ATP or don't form ATP. And then a synthetase requires energy to make covalent bonds. So this either uses or forms GTP in the Krebs cycle. And again, there's going to be another podcast that talks more about enzymes later on, so go listen to that. Enzymes are a very important topic. So now that I've talked a little bit about the enzymes, how to remember those, in the citric acid cycle, I want to go ahead and talk about the energy producing steps in the Krebs cycle. So as I talked about before, with glycolysis, you're going to want to know the energy producing steps and what form of energy is being produced, either GTP, ATP, or NADH, which can then be used to produce energy in the electron transport chain. So when I talk about energy producing steps, I'm going to talk about this in terms of the enzymes in which that energy is produced, or the enzymes that basically produce this energy molecule. So the first is Isocitrate Dehydrogenase, and that produces NADH. The second is alpha-ketoglutarate dehydrogenase, and that also creates NADH. And then you have succinyl-CoA synthetase, which creates GTP. And you have succinate dehydrogenase, which creates FADH2. And then lastly, you have malate dehydrogenase, which produces NADH. And later on, when I talk about the electron transport chain, I'm going to talk about how these reduced molecules transfer their electrons and then produce ATP, which your body can use. And so again, those molecules are NADH and FADH2. All right, so the last thing that I want to talk about in terms of the Krebs cycle is regulation of the Krebs cycle. So, what molecules kind of stimulate the Krebs cycle, what molecules inhibit it, and so forth. And this is very important to understand for the MCAT. And right now, I bet you $10 you will see some kind of regulation question, either a question about glycolysis or the Krebs cycle. So pay attention. So the Krebs cycle is regulated at 4 different points. The first point is in the conversion of pyruvate to acetyl-CoA. And remember this is the pyruvate dehydrogenase complex that has 3 different sub-enzymes that regulate its function. So the pyruvate dehydrogenase complex is downregulated or inhibited by ATP, acetyl-CoA, NADH, or fatty acids. And so if you think about it, these are molecules that are high energy molecules. So if your body has a lot of these high energy molecules, it's going to want to say, okay, we don't need to do as much of the citric acid cycle. We don't need to do as much of the Krebs cycle in order to give our body more energy because we already got all that shit. So on the other hand, the pyruvate dehydrogenase complex is upregulated when you have high levels of AMP, CoA, NAD+, or calcium ion, which is calcium 2+. And again, you can think these are low energy molecules, and therefore your body's like, we need more. So those are the molecules that regulate pyruvate dehydrogenase complex. The second point in which the citric acid cycle is regulated is at the citrate synthase enzyme. And so this enzyme is upregulated by ADP, again a low energy molecule, and is downregulated or inhibited by NADH, succinyl-CoA, citrate, and ATP. These are all high energy molecules, and it's important to note that succinyl-CoA is one of the intermediates of the citric acid cycle. So that basically feeds back and downregulates this enzyme. The third place in which the citric acid cycle is regulated is at the enzyme isocitrate dehydrogenase. And so there's a little bit of a pattern here. It is downregulated by ATP, high energy molecule, and is upregulated by calcium and ADP, which is again a low energy or lower energy molecule that signal that your body's saying, okay, we need to produce more energy. And finally, the citric acid cycle is regulated at the enzyme that is known as alpha-ketoglutarate dehydrogenase. And so similar to some of the others, it's downregulated by succinyl-CoA and NADH, and is upregulated by calcium ions. So one important thing to remember is succinyl-CoA inhibits at this step here, which is the last step, alpha ketoglutarate dehydrogenase, and it also inhibits at the second regulation step, which is citrate synthase. And so, yes, there is a pattern at each of these steps, but you really do want to know the exact details at each of these steps. So listen to this over if you need to, but have that memorized. And just a little recap, a good place to start for memorizing is remembering that high energy molecules means you have enough energy. Those are going to downregulate the enzymes in the citric acid cycle. And then on the other hand, these low energy molecules, they stimulate the Krebs cycle in order to produce more energy for your body to use. All right, so where are we at in metabolism? So we've gone through glycolysis, we've converted glucose into pyruvate, and then we've converted that pyruvate to acetyl-CoA, fed that through the Krebs cycle, also called the citric acid cycle, and then through that process, there's been some high energy molecules created in the form of ATP, GTP, NADH, FADH2, and then so that's where we're at. What happens next? Well, all these high energy molecules feed into what is known as the electron transport chain. So in the electron transport chain, essentially what happens is you have these reduced molecules, reduced as in they have electrons, extra electrons, and they transfer these electrons, they become oxidized, and through this process, a proton gradient is built up, and then that proton gradient
Is used to turn the ATP synthase, which then generates ATP.
So I'm going to get into this process, talk about the different complexes that make up the electron transport chain, how many protons are pumped across the membrane at each complex, what are the electron carriers, and then how does the ATP synthase work. All right, so there are 5 complexes of the electron transport chain. The 5th is also known as ATP synthase. And I think earlier I called it ATP ACE, but it's a synthase, so don't confuse that. So complex 1, what it does is, I'm going to start with how the electrons are transferred. So in complex 1, an electron from NADH is transferred to coenzyme Q, and 4 protons are transferred across the membrane. And then moving on, complex 2, an electron is transferred from succinate to coenzyme Q, and these electrons originate from FADH2, and this adds to what is called the ubiquinol pool. It adds to the ubiquinone pool, which ubiquinone is also coenzyme Q, so it's just making more coenzyme Q to add to that pool. And in complex 2, no protons are transferred across the membrane. And then in complex 3, you're transferring an electron from coenzyme Q to cytochrome C, which is a protein that contains a heme group. And a heme group consists of a iron cation bound to the center of a warfarin ring. And basically it's able to pick up electrons and able to transfer those electrons. So that's what you need to remember about cytochrome C. And this complex 3, again, displaces 4 protons across the membrane. That's the most out of all the complexes. And complex 4 transfers an electron from cytochrome C to oxygen and produces water. And in this step, there are 2 hydrogen ions displaced across the membrane. And before I get into complex 5, which is known as the ATP synthase, I want to talk a little bit about the reduction potentials as you move along these complexes. So remember that a reduction potential corresponds to how easily something is reduced. Or in other words, how good is it at taking an electron? That's the way I like to think about it.
A high reduction potential for a compound corresponds to that compound being very good at taking electrons from something else.
And so if you think about it, these electrons are being transferred as you move down. So you have to have an increasing reduction potential as you move along these, as you go from 1 to 4 in terms of these complexes, because you gotta keep having something that's better at taking electrons, or else those electrons are just gonna be stuck with something, unless you have something that's better at taking, and then as you move up, something else that rips it away because it's better, and so forth. So reduction potentials increase as you move along these electron carriers. And a common question, you might see something like, what has the highest reduction potential out of all these electron carriers or compounds in the electron transport chain? So what you got to do is you just got to think about which one of these electron carriers is furthest along in this process. So let's say you're looking through and you see O2. Well, that's going to be the answer, because that is the highest. That's the last thing that's reduced in this process. That's the last thing that these electrons are transferred, when the electrons are transferred to oxygen to make water. All right, so moving on to complex 5. And most of the time you're going to hear this referred to as ATP synthase. And
ATP synthases use the proton gradient built up by other compounds to create ATP. So remember that I discussed the number of protons transferred across the membrane for each complex. This builds up the proton gradient. And remember that the proton gradient is built between the intermembrane space and the mitochondrial matrix. And so the intermembrane space is where these protons are being pumped to. So the intermembrane space is going to have a much higher concentration of protons than the mitochondrial matrix. This gradient contains useful energy, and as these protons flow through the ATP synthase rotates and creates ATP. Each rotation corresponds to a different state of that ATP. You can look into that more if you want, but I don't think it's that important to know.
And so, quick note, it's important to remember that these protons are pumped from the mitochondrial matrix into the intermembrane space. And so to really understand how the ATP synthase functions, you have to understand the different portions of the ATP synthase. So first there's the F naught, and I'm not sure if it's F naught or F0, but it's just F with a subscript of 0. And this is the membrane spanning portion, and so this acts as a ion channel for the protons. It enables these protons to flow from the intermembrane space back into the mitochondrial matrix. And then you also have the F1 region, and this is the part of the ATP synthase that directly drives ATP synthesis. And so hydrogen ions essentially enter the F naught portion of the membrane and cause it to rotate, and this builds up energy. And this energy is used to make ATP. And so it's important to remember that 3 of these hydrogen are used in order to create 1 ATP from ADP and a inorganic phosphate, which is just a P subscript I. And that's about as deep of a look into the ATP synthase as you need to know. The most important part of the electron transport chain that you should know is, number 1, how many protons are transferred across the membrane at each point, and then 2, the molecules in which the electrons are transferred from. So those are the 2 things that you want to know. All right, so to give you a brief synopsis of metabolism that I just talked about, you take your glucose, you split it up into a few different molecules. Eventually those become pyruvate. Pyruvate is then converted into acetyl-CoA through the pyruvate dehydrogenase complex. And then the acetyl-CoA is fed into the citric acid cycle, where you create a lot more high energy molecules and oxidize it all the way to oxaloacetate. And then once you have all these high energy molecules, they all feed into the electron transport chain, where they donate their electrons, or they give off their electrons along a series of different molecules. And this enables some complexes within the electron transport chain to pump protons across the membrane. This builds up a proton gradient, and the energy from this proton gradient is then used to make ATP via the ATP synthase. And so now that I've covered all the different parts of metabolism, it is important to, well, first of all, I didn't cover all the parts of metabolism. There's obviously a ton of reactions going within your body to produce energy. I just went over the main ones you should know for the MCAT. And real quickly, the last thing I want to talk about are, number one, the different yields from each step of the metabolism steps I talked about, and then talk about the conversion units. So, how many ATP is one NADH worth? How many ATP is one FADH worth? So number one, in glycolysis, there are 2 ATPs produced, and there are 2 NADHs produced. And then in the conversion of pyruvate to acetyl-CoA by the pyruvate dehydrogenase complex, you produce 2 NADH. And then in the Krebs cycle, you produce 2 GTP, 8 NADH, 2 FADH2s for every 2 pyruvate molecules. So if you remember, one glucose is equal to 2 pyruvate, so that's just the rates for 1 glucose molecule. So now I know you're definitely thinking, all right, what are these conversion units for these high energy molecules? Well, NADH goes for 2.5 ATP, and 1 FADH2 goes for 1.5 ATP, and then of course 1 GTP is equal to 1 ATP. So overall, after you've gone through all of these steps of metabolism, all the different cycles and everything, you end up with 30 to 32 overall ATP for 1 glucose molecule. Pretty good. So I hope you enjoyed, and I hope this helps. Thanks for listening.
All right, so now for the Mcat Advice of the day. In terms of practice materials, I always recommend the AAMC official practice materials, all the question packs and all the practice tests and stuff. Some of the other stuff that the other companies put out is good, but for the most representative stuff, look for the official materials, especially in terms of practice tests. So, save the practice tests for when you really want to know how you're going to do on the test, because those line up really well with the actual test. You'll score plus or minus a few points of what you get on the practice tests to the actual tests. And there's 3 official practice tests that are out by the AAMC. And they also put out a sample test that's not graded by the MCAT scale. It's graded on just, how many percent of questions you got right on a certain section. So that's not great in terms of telling you where you're at, but still useful for studying. And so if you want to take a bunch of tests, which you should, you're going to have to buy it from a third-party company. And so there's quite a few of them, but all the other study materials, try to really focus on the AAMC stuff, know that really well, and kind of just fill in, maybe pick another company. I honestly don't think it matters that much. Pick another company, go through their review stuff, but don't buy all the books from every company and think that you're going to get through all of it, because number one, you're not, and number two, it's not necessarily going to be representative of what you see on the test.
Come on, Murph. I'm trying to podcast over here, dude.